Okay, it's great to be in Estoril, Portugal, at the European School of Hematology meetings. And I'm interested in the immunology of AML and whether it's a disease which is susceptible to immunotherapy. So we think that there are some significant challenges for the immunotherapeutic approaches for the treatment of AML, and those include the lack of very good targets to go after. The targets that are used and are focused on by our group and others are also expressed on mature myeloid cells, and that creates a large sink of target which you have to get past to get to the AML cells, so that's a problem, the extent of this sink. The second is that the target's expressed on not only AML cells but also on mature monocytes and granulocytes. And monocytes and granulocytes are the cells that actually make a lot of the cytokines that cause what's called cytokine release syndrome, and that's associated with sometimes life-threatening complications such as fever, chills, hypotension, etc. So in order to really target AML, you have to get past the sink and you have to reduce the CRS-related toxicity. So that's a problem. The final issue is that the targets are also expressed for the most part on hematopoietic stem cells, and that means that if you're successful in targeting AML, you might also be successful at targeting the hematopoietic stem cell, and that would be a problem because you need blood cells to survive. And so those are all potential complications and issues relating to the targeting of AML. There are no really specific selective targets that are uniquely expressed on AML and not on other cells, and so it requires some sort of thinking outside the box. So one approach would be to try to develop methods to enhance the sensitivity and the efficacy of immunotherapy so that you can use it at lower doses so that you don't have to deal with this CRS. And people are doing that by using what's called checkpoint inhibitors, and we're interested in that as well. But more importantly, we think that another approach might be to advance the dose of these immunotherapies at higher doses that are normally tolerated by blocking the CRS. That's called CRS mitigation approaches. And by doing that, we can actually block some of the inflammatory changes, and that will allow us to advance the dose of these drugs so that they're more effective. And the third approach would be to develop completely novel ways of enhancing the immune effects of bispecifics such that perhaps at the same barely tolerable dose they're much more effective, and so we're interested in that as well. And one of the ways that we're doing that is by using hormones that actually, not hormones, but cytokines that extend the life and survival of the T cells which are engaged with the AML cells. So by doing that you can enhance the ability of those T cells to kill the targets and eliminate the AML.